For decades, we've been trying to build computers that think like human brains. But what if we've been going about it all wrong? What if instead of that, we could actually teach human brain cells how to be computers? Well, today, that's exactly what we're talking about. We're diving into the incredible new field of organoid intelligence, where scientists are, and I mean this literally, teaching lab-grown clusters of brain cells how to compute. Okay, you really have to let this one sink in for a second. 40 minutes for 1 second of just 1% of brain activity. This single fact is just staggering, right? It shows you the almost unbelievable power of biological computation. The brain's efficiency is just on another level, and that's the inspiration for this whole new kind of radical approach to computing. For as long as I can remember, the big quest has been to make silicon chips act more like our brains. But what if that's the scenic route? What if we could just skip all that and tap directly into the real thing? This is where the entire script gets flipped. So, welcome to the world of organoid intelligence, or OI for short. The core idea is actually pretty straightforward, but wild. We take these 3D cultures of brain cells, they're called organoids, and we use their own amazing self-organizing power to teach them. Teach them to remember things, to process information, to become a living computer. And this right here is the key difference. You see, artificial intelligence, AI, is all about making machines think more like us. Organoid intelligence, OI, turns that completely on its head. It asks the opposite question. How can we get a culture of living brain cells to think more like a computer? It's a total paradigm shift. So how on earth do you even begin to teach a tiny clump of neurons anything? I mean, you can't just plug in a monitor and keyboard, right? Well, the challenge is this amazing mix of biology, some really clever engineering, and even a little bit of artificial intelligence. Okay, so before a brain organoid can even think about learning, it has to be alive and complex enough to do the job. And here's the first big problem. As these things grow, the cells in the middle start to starve. They just can't get enough food, so the core literally starts to die. And the solution is it's a work of art. Engineers built these incredible microfluidic systems that act like a tiny artificial circulatory system. You've got these little channels pumping nutrients deep inside and pulling all the waste out. It's what allows these organoids to get bigger, more complex, and a whole lot tougher. All right, so the organoid is alive, it's growing, it's healthy. Now what? We need to have a conversation with it. How do you ask it a question? And maybe more importantly, how do you understand its answer? This is where we need a brain-machine interface. The answer is this amazing piece of biotech. They're called 3D microelectrode arrays. Just picture a tiny, flexible shell that's absolutely covered in electrodes. The organoid grows inside of it, and this thing fits around it like a perfect little glove. It lets scientists send electrical signals in and read the neural signals out with incredible detail. It's basically a tiny EEG cap for a mini brain. So, here's where it all comes together. Check out this training process. It's this really elegant feedback loop. First, you stimulate the organoid with an electrical signal. That's the question. Then, you record the electrical activity that bounces back. That's the answer. Now, here's the magic part. An AI decodes that super complex, noisy answer, and then, this is the crucial step, the AI gives feedback, changing the next question to slowly guide the organoid toward the right kind of response. It's an AI playing the role of a personal trainer for a biological brain, pushing it in the right direction, one pulse at a time. Okay, this is all unbelievably cool tech, but we've got to ask the big question. Why? What's the end game here? What does teaching a bunch of cells to compute actually do for us in the future? Well, the potential is just huge. I mean, for one, we're talking about biocomputers that could be way faster and incredibly more energy efficient than anything we have today. But honestly, it goes so much deeper than that. This is a brand new way to study the human brain itself. We could model diseases like dementia, test new drugs on actual human brain tissue without harming anyone, and maybe, just maybe, finally figure out how things like memory and learning actually work. And just to give you a little perspective on the kind of power we're talking about, that's the estimated storage capacity of one human brain. 2,500 terabytes. Your laptop probably has, 1 terabyte? Maybe 2 if you're lucky. This is the biological hard drive we're just now learning how to plug into. Now, obviously, you can't start growing and teaching human brain cells in a petri dish without bumping into some really profound, really complicated ethical questions. This research forces us to look at some of the deepest questions we have about what life and intelligence even are.
And here it is. This is the elephant in the room, right? It's the one question that hangs over this entire field. As these things get more and more complex, as they get better at learning, where do we draw the line?
To even have this conversation, we need to be really clear about our words. Researchers make a super important distinction here. There's sentience, which is just being able to sense something and react to it. A bacteria can do that. But then there's consciousness. That's the subjective feeling of being, the what it's like to be me feeling. Right now, the goal is basic sentience, not consciousness. This leads to a really tricky paradox called the Greeley dilemma. Basically, the better scientists get at modeling the brain to, say, cure Alzheimer's, the closer that model gets to the real thing, and the bigger the ethical questions become. Every single success pulls us deeper into this dilemma.
So what's the plan? Well, the good news is the scientific community is taking this incredibly seriously. The solution is something called embedded ethics. The idea is to have the ethicists, the scientists, and even the public all in conversation together from day one. It's about making sure the science doesn't race ahead of our ability to handle it.
And that leaves us with one last massive question to think about. As we keep blurring this line between a computer chip and a neuron, between writing code and actual learning, where do we, as a society, draw the line between a very powerful biological tool and a being?